Okay, I hope you guys already have a clear picture about biocontrol in the soil and how human activities in general and agriculture practices, to be specific, can change the paradigm of soil food web and or specific antagonists to suppress the pathogens in the soil. Okay, uh, here I will give some case studies, especially that related to biocontrol in agriculture. So the first case study is the Phytophthora suppressive soils. Okay, so this Phytophthora cinnamomi is a widespread soil-borne fungal pathogen that infects woody plants, causing root rot and cankering. Okay, so there is one research on avocado groves in Queensland, Australia. There is a severe and economically significant outbreak of root rot due to this fungi, this Phytophthora cinnamomi. Okay, this is the first documented in 1974 by Broadbent and Baker. So researchers investigating this disease outbreak discovered that suppressive soils in a neighboring okay, avocado grove that exhibited only minor damage from root rot. Okay, so therefore they introduced the soil from the neighboring field to the uh, to the to the new field. Okay, so of course this management practices promoted the growth of an active soil microbial population. So this effect suggests that spore forming bacteria and actinomycetes were the biological antagonists acting against the Phytophthora fungal pathogen. Of course, no single organism was capable of suppressing the fungal pathogen, which led the researchers to conclude that the control mechanism was the general suppression. So this is for the first case study on the Phytophthora suppressive soils. So again, how the scientists discovered, they found that the soil from the neighboring field has less problem with the root rot. So they introduce the soil into the new field, and they can control the root rot in the new field, which means that there is some activities of soil microbes from the field in the previous one, in the first one with no root rot, that help in fighting these fungi. Okay. Next is the second study is the suppression of take-all disease in wheat. Okay, this take-all disease of wheat is caused by the fungus Gumanomyces graminis. Okay, this root infection by the fungus blocks the conductive tissue and reduces water uptake. So, in many parts of the world, producers gain partial control of this take-all disease, by just rotating wheat and barley crops with other non-host crops. However, the disease is transferable, okay, from field to field, so it can be spread by missionaries, for with human by in the large areas. Okay, there are two biological controls. Weller in 2002 suggested two biological control mechanisms that can reduce take-all severity in wheat. First is by competition from bacteria and fungi in root fragments, which reduces the inoculum density of the pathogen, this is a general suppression. And secondly, they also can introduce antagonist microorganisms in the rhizosphere, and young root lesions attack the pathogen and limit its infection and spread through roots, which means that this is a specific suppressions. So general suppression of the take-all pathogen is partially attributed to the activity of non-pathogenic saprophytic Fusarium, okay, Fusarium species. The take-all pathogen is able to grow and reproduce on crop residues like such as in root fragments and stems when the crop is not present in the field. The fungus Fusarium, okay, the fungus Fusarium here, okay, the fungus Fusarium, what we call as is more efficient at obtaining energy and nutrients from decomposing residues than the take-all pathogens, thus reducing the amount of the Gumanomyces graminis inoculum that could infect the next wheat or barley crop. Specific suppression also can be achieved by antagonist fluorescent Pseudomonas. This specific, which is an important means on biological control. So this what we call as the specific Pseudomonas called Pseudomonas fluorescens. The most biological control strains of Pseudomonas, the Pseudomonas species produces the 2,4-DAPG. This is an antibiotic synthesized by the Pseudomonas that live in the rhizosphere and is effective at reducing the growth of the take-all pathogen. So this is a clear example of specific suppression by Pseudomonas fluorescens. Okay, so this is for the take-all disease in wheat. Okay. Moving on, the third case study that I want to share with you guys is the suppression of root knot nematode, Meloidoigene species. Okay, this is the nematode species. These root nematode are serious pests in crop production because they infect a variety of crops including tomato, banana, grapes, carrots, strawberry, lettuce, pumpkin, and even bean. Okay, damage to the root, like we show here. Okay, caused by nematodes reduce the root capacity to absorb water and nutrient necessary for crop production. So biological control of this nematode can be achieved through specific suppression with Bacillomyces liliaceus. Okay, this Bacillomyces liliaceus, this nematophagous fungus capable can reduce root knot population by 76%. The another one, the nematode trapping fungus Monacrosporium lysipagum, can reduce root knot nematode by 93%. So this is clearly the specific suppression. So of course, the general suppression of root knot nematode is also possible and relies on crop rotations and cover cropping with legumes. So intercropping also can intercropping of susceptible crops and non-host crops is another technique that has been used with some success. Okay, so here are some of the examples of case studies that use biological control, use biocontrol to suppress pathogens in the field.